All right, diving in today, and I'm excited about this one. We're going deep on something most people probably don't think about much, but it's really, really important. Impurity profiling and how those are controlled.
It's kind of behind the scenes, but yeah, super important.
Exactly. And you sent over a really cool collection of sources, stuff on drug development, the actual manufacturing processes, and of course the regulations.
Mm.
Can't forget those. What we're aiming for today is to break down, really get to the nuts and bolts of how companies make sure the meds we all take are safe. And a huge part of that is finding and dealing with any impurities. So we're going to dig into the methods they use, look at some real-world examples, give everyone listening a good grasp of this whole world that's kind of hidden within drug production.
It's like you get your medicine, you trust it's going to work, right? But behind that trust, there's this whole system making sure what you're swallowing is exactly what it should be and nothing it shouldn't.
That's a great way to put it. So much goes into it that we never see. And honestly, kind of scary to think about what could happen if they didn't do this.
Oh, absolutely. You hear about those rare cases where there's a contamination issue.
Yeah.
That's why this whole impurity control thing is not just like an optional extra. It's really fundamental. It's about safety, plain and simple. And that's where those regulations come in. They're the backbone of this whole thing.
They drive the whole process.
Totally. And as we talk about specific examples, you'll see exactly how those rules shape what manufacturers actually do.
Okay, so let's get down to it. How do scientists even find these impurities, these like tiny little unwanted bits? I mean, we're talking microscopic level here.
Yeah, it's pretty amazing.
Right. So what are the tools? What are they using?
Well, one that popped up a bunch in what you sent, especially when they were talking about chiral drugs, was HPLC, high-performance liquid chromatography. Sounds intimidating, but
Super precise.
yeah.
From what I read, it's like the ultimate sorting machine.
That's a good way to think about it. It's like you have this complex mixture, right, your drug sample, and HPLC can separate out each individual component, and not just separate them, but measure them too. So you know exactly what's in there, the good stuff, the active ingredient, but also any impurities lurking around. And with chiral drugs, it gets even cooler.
Which, just to remind everyone, those are the ones that exist in two mirror image forms, like our hands, a left and a right version.
Exactly. And sometimes only one of those forms is the actual medicine, the effective one. The other one could be totally useless, or even worse, cause problems.
Could even be harmful.
Right. So HPLC can tell them apart. That's why it's so powerful in this context, that ability to do such specific separation.
So crucial.
Mhm.
Makes you realize it's not just about what's there, it's about the specific version of it too. Now, another technique that came up was NMR, nuclear magnetic resonance. Now, I always picture that as like a way to map out the structure of a molecule. Is that right?
Yeah, you got it. NMR is fantastic for figuring out that whole molecular architecture, how all the atoms are linked up, the whole blueprint. But here's where it gets even better for impurity profiling. We're not just using NMR to ID what the impurity is anymore, but also to say exactly how much of it is present.
So it's about quantifying it.
Exactly. It's becoming super reliable for measuring those unwanted bits. So you get the ID and the quantity all in one go.
It's amazing. Two birds, one stone. And often working hand in hand with NMR, we have mass spectrometry, or MS. I remember reading that together. They're like the dynamic duo for unraveling the mysteries of unknown impurities.
They really are. It's like
Yeah.
NMR gives you the blueprint, right? And then MS comes in and basically it weighs the pieces of that blueprint. By looking at those weights, we can figure out what an impurity is, even if we've never encountered it before. And when you use high-resolution MS, you get crazy accurate ID. One of the sources you sent talked about LC-HRMS, which is just liquid chromatography hooked up to high-resolution MS.
That's a mouthful.
I know, right? But it's incredible tech. And they used it to identify and measure NDMA in ranitidine. NDMA, this nitrosamine impurity, it caused a whole bunch of recalls and investigations.
That was huge news a while back.
It was. And it just goes to show you how powerful and important this kind of impurity profiling can be. It's not just science for the sake of science. It has real-world implications.
For sure. For sure. Now, shifting gears a bit, let's talk about another kind of impurity that might be lurking. Elements. For this, it seems ICP-MS is the tool of choice. Inductively coupled plasma mass spectrometry. So we're talking about things like detecting metal residues or other elemental stuff.
Spot on. ICP-MS is the go-to for measuring elemental impurities in drugs. And these could come from various sources. Maybe there was a metal catalyst used during synthesis and some of it stuck around. Or maybe it's naturally occurring elements that were in the starting materials.
Oh, so it's not always like contamination in the factory sense.
Not necessarily. Sometimes it's just like inherent to the stuff they're working with. But the challenge with ICP-MS is that you often get interferences. This means one element, or ion, can mask the signal of another one because they have similar masses.
Like trying to pick out a single voice in a crowded room.
Exactly. A classic example is argon, which has an atomic mass of 40, interfering with calcium, which also has a mass of 40.
So how do they differentiate? How do they tell those apart?
Well, they have some clever tricks up their sleeves. ICP-MS instruments often have collision or reaction cells to deal with this problem.
Okay, break those down for us. What are those?
Sure. So in a collision cell, they introduce a low reactivity gas, something like hydrogen, into the path of the ions. This gas collides with the ions, and because interfering ions tend to be a bit bigger or have a slightly different charge, they lose more energy in these collisions compared to the analyte ions, which are the ones we want to measure. And that energy difference can be used to separate them out. The downside is that the analyte ions can also lose some energy, so our ability to detect really, really low levels might be slightly reduced. Reaction cells, on the other hand, use reactive gases like ammonia to selectively react with the interfering ions, changing their mass so they no longer overlap with what we're trying to measure. But here, the challenge is making sure that reaction is super specific so it only affects the interfering ion and not the analyte.
So a lot of fine-tuning.
Yeah. The choice between these approaches depends on a bunch of factors, but ultimately it comes down to which one gets rid of the interference and still lets them detect the impurity at the level they need. One of the sources mentioned that for calcium detection, a reaction cell using ammonia is way more sensitive than a collision cell with hydrogen, simply because it does a better job at eliminating that pesky argon interference.
So it's not just about, does it work? It's about, does it work best for this particular analysis?
Exactly.
Wow, it's amazing how much complexity goes into just figuring out what's in these meds, even at that tiny scale.
It's mind-boggling for sure.
So once you've got all these fancy tools to pinpoint potential impurities, what happens next? How do manufacturers actually control them during the whole manufacturing process?
That's where the rubber meets the road. It's where the science of chemistry meets the practicality of engineering. And it's all about designing processes that build in controls to minimize the formation of impurities from the get-go, and of course remove any that do pop up.
So it's kind of a two-pronged approach, prevention and cleanup.
You got it. And process optimization is key here. It's all about tweaking the conditions to get the best outcome. One of the OPR&D sources had this great example where they were making a certain compound and they had this impurity, they called it bisform impurity D, that was forming during the synthesis.
What kind of tweaks are we talking about?
Well, in this case, they played around with the amount of DMF, which is a common solvent, and the speed at which they added another ingredient, carbonyl imidazole. And by carefully adjusting those two things, they were able to dramatically reduce the amount of that pesky byproduct.
It's like a recipe, isn't it? Like changing the order you add ingredients or how much you use can totally change how the dish turns out.
Exactly. And crystallization is another key player in this control game. It's often thought of as just getting the drug into a solid form, those nice little crystals you see. But it's actually a really powerful way to purify things, too. There was this really cool example in one of the sources about how they use different solvents like IPAC and methanol to improve the optical purity of a salt form.
Remind us again, optical purity meaning.
Right. So that means making sure you have the right proportion of the two mirror image forms. Like if you only want the left-handed version, you want to make sure there's not too much the right-handed one mixed in. And what they found was that this particular impurity, a hemisalt, was forming and messing up their attempts to get that pure drug they were after. But by picking the right solvent combo, they could essentially dissolve away the hemisalt and crystallize out the pure form. It's like using solubility to do your dirty work.
Neat. So you're not always trying to react the impurity away. Sometimes you can just kind of dissolve it away.
Yeah.
We also saw stuff about using scavengers to remove specific types of impurities.
Oh yeah, scavengers are super useful, especially when you use metal catalysts in the synthesis, like palladium, which is pretty common. You got to make sure none of that metal ends up in your final product. And that's where scavengers come in, these substances that basically grab onto the metal and pull it out.
They just like snatch it up.
Pretty much. Activated carbon, like charcoal is a common one. It just absorbs a bunch of stuff. But they also use more specific chemicals like TMT or L-cysteine, which is an amino acid.
TMT being...
Oh, right. Sorry, 2,4,6-trimercaptose triazine.
That's a mouthful.
Tell me about it. But anyway, the interesting thing they talked about was using L-cysteine because it could avoid having to do another filtration step to get rid of the activated carbon. And that's important because from a good manufacturing practice or GMP standpoint, They're always worried about potential contamination from the carbon itself getting into the final product.
You don't want to solve one problem just to create another?
Yeah. Exactly. So even the choice of scavenger isn't just about whether it works or not. It's about all these other factors like practicality and safety.
Absolutely makes sense. Now, another technique that sounded super cool, almost futuristic, was continuous flow chemistry.
Oh, yeah. That's definitely a hot area in pharma manufacturing. Instead of doing reactions in big batches, they use this like mini chemical factory, a network of tubes where they pump the reactants through.
So like a little assembly line.
Yeah, kind of. And because of that, they can control all those reaction parameters super precisely. Temperature, pressure, how long the ingredients are reacting. And that fine-tuned control can really cut down on those unwanted byproducts, leading to a much purer product right from the start. One of the articles you shared mentioned using continuous flow for making these complex molecules where timing is everything.
Because if the timing's off, you get the wrong stuff.
Exactly. And that precision is what helps them avoid those impurities that might pop up if the reaction isn't perfectly timed.
It's fascinating. So much more control than just dumping everything in a big vat and hoping for the best. And then to make sure things are on track, there are these in-process monitoring techniques, right?
Absolutely. They're like having little checkpoints along the production line. Techniques like thin layer chromatography or TLC, which is a quick and easy way to check what's in a mixture, gas chromatography or GC, which separates things based on how easily they evaporate, and even NMR can be used to monitor the reaction as it's happening. This way, scientists can see right away if any impurities are starting to form and make adjustments to the process before things get out of hand. It's all about keeping a close eye on things.
Makes total sense. All right, we've covered a lot of different methods, and it's been super interesting, but let's bring it back to the real world for a second.
Yeah.
Those OPR&D sources you sent over had some great case studies that really show how this all comes together.
Let's talk about them.
Okay, so remember that enantiomeric purity upgrade we were talking about earlier, where they had that hemisalt forming and messing things up?
Oh yeah, the one where they figured out the right solvent combo.
That's the one. They didn't just stumble on that solution, it was systematic. They really dug deep into understanding the chemistry, the properties of the drug, and the impurity. And they even used NMR to confirm the structure of that hemisalt, which helped them figure out which solvents to use. It's a great example of how this isn't just trial and error, it's about using analytical thinking to guide the process.
Totally. And the one about using L-cysteine to remove palladium, that was a good one too. They specifically chose L-cysteine over activated carbon because remember, they were worried about carbon contamination and needing an extra filtration step. It really highlights how GMP considerations are woven into every decision they make. It's not just about the chemistry working in a lab setting, it has to be safe and reliable for large-scale production.
Can't cut corners when people's health is on the line.
Exactly. And then there's that other example where they tweaked the synthesis to reduce that biform impurity D. They started with a method that used a lot of DMF, which was a pain to remove, not very practical for large-scale production. But by changing the rate they added one of the reactants and optimizing the solvent system, they got the impurity levels way down consistently. That kind of process refinement, it's crucial for making sure you have a drug product that's high quality, batch after batch.
Consistency is key.
Definitely.
And it's so clear that all these methods, all these control strategies, they're not just some theoretical stuff. They're directly tied to meeting those strict safety standards set by regulatory agencies all over the world.
Couldn't agree more. And even though we didn't get into the nitty-gritty details of those regulations today, it's important to remember that they drive everything in this field. They're not guidelines, they're requirements. And if manufacturers don't identify and control impurities properly, the consequences can be serious.
Right.
We're talking about the effectiveness of the drug and, even more importantly, patient safety. So these efforts, all these details we've been talking about, they're the foundation for making sure the medicines we all depend on are as safe as they can be.
Well said. So as we wrap things up, what are the big takeaways, the things you really want people to walk away with from this deep dive into the world of impurity profiling and control?
I think the main thing is, I hope they have a better appreciation for the complexity, how much goes into making sure our meds are pure. It's not just one thing, it's layers and layers. You've got these incredible analytical tools to find even the tiniest amounts of unwanted stuff. And then you've got this whole science of designing and controlling the manufacturing process to prevent those impurities in the first place or get rid of them efficiently. And those real-world examples, those case studies we talked about, they really bring it all home, show how these principles are put into action day in and day out to make sure the medicines that people rely on are safe and effective.
It's been a real eye-opener for sure, and can leave everyone with something to think about. A question. With all the amazing advancements in technology, both in the analytical tools and in manufacturing itself, how do you think our ability to find and control impurities, even at those incredibly tiny levels, is going to evolve in the future? And how might that impact drug safety? What about developing totally new kinds of therapies? It's exciting to think about.
There's so many possibilities.
Right. It's a fascinating area for sure. Thanks for joining us for this deep dive.
My pleasure. Always fun to geek out about this stuff.